We study today in embryology structure of anther. In the structure of anther, as seen in this flow chart, anther structure divided in two parts: outer epidermis and archaeosporium. Archaeosporium again divided in parietal tissue and sporogenous tissue. Parietal tissue have endothelium, middle layer, and tapetum. Sporogenous tissue mainly function of this type of tissue formation of pollen. In a flower, stamen is considered as the male reproductive organ. Each stamen consists of filament, connective, and anther. Anther may be monothecous or dithecous. A monothecous anther consists of two locules or two sporangia. So it is said to be bilocular or bisporangiate. A dithecous anther consists of four loculus or four sporangia. So it is said to be tetralocular or tetrasporangiate anther. Development of microsporangium is eusporangiate type. Sporangium developing from a group of cells. A very young anther in transverse section shows epidermis and archaeosporium. The archaeosporial cells divide periclinally, giving rise to primary parietal cells on the outer side and sporogenous cells towards the inner side. The cells of parietal layers divide periclinally and anticlinally, forms endothelium, middle layers, and tapetum. The cells of the primary sporogenous tissue differentiated into pollen mother cells and microspore mother cells. The anther wall consists of following wall layers divided in four parts: epidermis, endothelium, middle layers, and tapetum. First, we study epidermis. Epidermis is the outermost single layer. It is compactly arranged and usually protective in function. Epidermal stomata was reported in Elangium. Endothelium. The cells of endothelium are radially elongated and shows fibrous bands. The fibrous bands are made of a callus and arise from inner tenacious walls. Usually, fibrous bands are U-shaped. Fibrous bands are hygroscopic in nature. Endothelial cells helps in the dehiscence of anther at maturity. The main function of endothelial cells. Because of the presence of fibrous band, this layer is otherwise called fibrous layer. It is usually single layered. Middle layers. Below the endothelium, 2 and 3 layers of cells are present, which constitute middle layers. They are known as middle layers. These layers are ephemeral in nature and become crushed by early meiosis in pollen mother cells. The cells act as a storage center for starch. Tapetum is the innermost layer of anther walls and it completely surrounds the sporogenous tissue. The cells contain dense cytoplasm with prominent nuclei. Usually tapetum consists of single layer of cells. As the tapetum completely surrounds the sporogenous tissue, major part of it is derived from the parietal cells and a small part developed from the sporogenous tissue. Tapetum transports the nutrients to the developing sporocyte. This is the major function of tapetum. Tapetal cells are pigmented and it is red-brown in apple or violet On the basis of behavior, there are two kinds of tapetum. Tapetum divided in two types. First one is amoeboid tapetum. The inner and radial walls of the tapetum break down due to the action of hydrolytic enzymes, and their protoplast penetrates between the pollen mother cells and developing pollen grains. After intrusion, they fuse with each other and forms a mass of tapetal periplasmodium. This type of tapetal plasmodium remains associated with the pollen grains till their maturity. When the anther gets drying up to the tapetal periplasmodium gets dehydrated and coated over the surface of pollen grains, thereby helping in the formation of exine. Amoeboid tapetum is considered as the primitive type of tapetum. It is also called periplasmodial tapetum. Example: Tradescantia, typha, Sagittaria, potamogeton. Next type of tapetum is glandular tapetum. The cells of glandular tapetum remains intact throughout microspore development. They secrete their substances from their inner facade. Secretory tapetal cells are thin and possess almost all cell organelles like mitochondria, plastids, dictyosomes, etc. Just before the pollen mother cell undergo meiosis, the walls of the tapetal cells become thick and there is considerable increase in the number of ribosomes and pro-Ubis bodies. With the completion of pollen development, pro-Ubis bodies pass into the anther locule from the tapetal cells, and they are now called Ubis bodies, and they coated over the pollen grains. Example: higher monocots and many dicots. Functions of the tapetum: the nutrients are transported through tapetum to the sporogenous tissue. Tapetum is involved in the synthesis of callose, which release microspores in a tetrad by degrading callose wall. Tapetum plays an important role in the formation of exine of the pollen. Pollen kit lipids. Pollen kit is made from lipids and carotenoids. It is also formed by tapetal layer. It is an insect attractant and protect pollen from ultraviolet rays. Here is the transverse section of tetrasporangiate anther with showing the various tissues. In the center part, a connective tissue as we seen. The outer boundary is epidermis. This one is followed by endothecium, then middle layers, and last the pollen grains surrounded by tapetum cells.